it's my great pleasure today to introduce Dr. Nicholas Shork, who is visiting us from San Diego. He's the Director of Biostatistics and Bioinformatics at the Scripps Translational Science Institute, which is an independent research institute, very much like MRN, just 10 times as large. And he's also the Director of Research for Medicine and Genomics, and a professor at UC San Diego. He has, and a member of numerous advisory boards and special panels and all that good stuff, and holder of many patents. And he's been working for several decades in genetics and methods of genetics analysis, combining statistics and epidemiology and philosophy originally, which I can understand. There's a certain amount of philosophy to all of this. He's really been contributing in key ways to the development of genetic methods over the last couple of decades. He has broad expertise in many diseases and experimental designs. He's known for his creativity and innovation. We're just honored to have him here and looking forward to hearing about his results on a topic near and dear to my heart, polygenic effects on the brain. So, thank you, Nick.
Great. Thanks for having me. I was really flattered to be asked to come. Jessica and I have interacted over the years, first when she was in Orange County, and now most recently, obviously, being in Albuquerque. What I'd like to do is kind of walk you through some things that my group has been working on for about the last 4 or 5 years, having to do with understanding how genetic variation impacts phenotypes of all sorts. Today I'm going to focus on brain imaging phenotypes, or phenotypes derived from, say, MRI. In order to do that, though, I'd like to put some of what I'm going to talk about later into a context. So the first 10 minutes or so of my talk, I'm not going to give you any insights into genes that might influence the brain, but just the role that genetic diversity has in, or the potential impact that genetic diversity has in the use of genetic assays in clinical practice. So I'll start with giving you a brief synopsis of what is available in terms of genomic assays. You probably know all this anyways. Talk about genetic background and its impact on the identification of variants that influence what are termed idiopathic conditions. And some of you are probably familiar with what an idiopathic condition is. And then talk about genetic influences on brain morphology based on imaging genetic studies. Give you a little bit of a rundown on a recent paper that we published looking at brain morphology and its correlation with genetic background among Europeans. Talk about genetic background and global variation at a much broader scale using a data set derived from the PING study, the Pediatric Neuroimaging and Genetics Study, led by Anders Dale and others at UCSD that you might know about. And then talk a little bit about other studies, maybe a couple of slides, in particular focusing on genetic influences on scaling phenomena in the brain, and then end with a few conclusions and possible future directions. Okay, so these pictures right here are of people who suffered from an idiopathic condition, a condition that was so rare it only seemed to manifest itself in a single individual. Okay. Now, with the exception of Jim Lupsky, who has a unique form of CMT, the other people had conditions that defied easy diagnosis. Nicholas Volker probably being the most famous and considered sort of a poster child for success that genomics has had on yielding insights into the pathogenic basis for an idiopathic condition. So Nicholas Volker, just to give you a little bit of background on what idiopathic conditions are and why he's considered the poster child, had an inflammatory bowel-like condition up in Milwaukee that none of the docs could figure out how to deal with. It didn't show the sorts of clinical manifestations that typical inflammatory bowel disease-like conditions showed. In order to actually treat the pathology that Nicholas Volker had, he actually had to have surgeries to kind of go in and clean out the gunk in his gut. And he had over 100 before he was 5 years old. And there's no other way of treating the disease. They tried everything in the kitchen sink, nothing worked. He actually maxed out on the total allotment he had from his health insurer, $2 million to pay for those surgeries. So his family was faced with this dilemma of no longer being able to rely on health insurance, and yet knowing that the only way they could treat his condition was to have these continued surgeries. So his parents approached actually a colleague of mine up at the Medical College of Wisconsin, Howard Jacob, and said, well, could you just sequence Nicholas Volker's genome? The belief being that maybe they'd identify some big deletion of a gene that could shed light on what is causing this condition and maybe come up with a therapy. Knowing that this was a real flyer and it might not work, Howard decided to do it anyways. They sequenced initially his exome and identified a mutation in a particular gene that had never known to be mutated in an inflammatory bowel-like condition. But there were people that had mutations in this gene that suffered from conditions that, again, didn't look anything like the sort of condition Nicholas Volker had, that were treatable with a bone marrow transplant. Now, no one would have thought to treat his condition with a bone marrow transplant, but given that there was a mutation in a gene, a different mutation than what other people had, but still the same gene, where those other mutations and the pathologies they induced were treatable with a bone marrow transplant, they decided to go forward with the bone marrow transplant. And in fact, they did, and now he is perfectly healthy, playing t-ball, eating regularly, and not having the surgeries. And in fact, the reporters at the Milwaukee Sentinel, I think is the newspaper, who covered this story actually won a Pulitzer Prize for their depiction of the ordeal that the family was faced with and what they went through in terms of sequencing his exome and coming up with insight into his disease. The Beery twins are from Carlsbad, just about 20 minutes north of where I am in La Jolla, had a similar sort of condition that defied easy diagnosis. Their genomes were sequenced. They were found to have unique variations in dopamine and serotonin genes. They've now been treated with particular drugs. And are doing fine and now playing volleyball, though when they were children, they were essentially confined to wheelchairs. So similarly with other congenital conditions that defied an easy diagnosis, variants were identified that shed light on these conditions. So just to recap, idiopathic conditions are those that defy conventional diagnostic categories and are typically treatment unresponsive. So every hospital system has some number of patients that come in that nobody quite knows what to deal with. Sequencing the genomes of individuals with idiopathic conditions could shed light on the origins. And again, if you've tried everything else, then why not sequence the genome? That's sort of the going thing with sequencing in idiopathic conditions. The variants that influence these diseases could be inherited in quite complex ways, like through compound heterozygosity, or the mutations could be de novo. In fact, many are thought to be de novo because no one else on the planet has conditions similar to the ones that these patients manifest. Finding the pathogenic or causative variant among the many candidates in the genome, given the size of the human genome, 3.2 billion nucleotides, any one of which could be perturbed to cause a disease, this is a big kind of needle in haystack-like problem. Strategies based on whole genome sequencing and the use of reference genomes and bioinformatics tools, however, exist to kind of identify from all the variants that could be candidates those that are most logical. And in fact, those tools were used to identify the variants responsible for these people's conditions. So, how does that strategy work? Well, it's based on kind of a filtering strategy. You start out with all these variants and you narrow it down into the ones that you think are the most likely causal and pathogenic variants. Makes two basic reasonable assumptions. I have a question mark there. You could challenge these. First is that the pathogenic variant is novel. That is, it's more or less unique to the genome of the individual with the disease. The second one is that the effect of the variant is pronounced enough to be characterized through computational techniques. Now, why is that important? Because if you're looking for a variant that no one on the planet has ever seen before, it's probably the case that that variant hasn't been subjected to all sorts of functional assays that then people can use to label that variant as likely damaging or doing something that could cause a disease. So what you might want to do is take all the variants that you think are candidates and at least make predictions on whether they might be deleterious or have a functional impact. And you can do that with computational tools. So the way this filter works is you have your patient and you identify or sequence his or her genome and identify variants, non-reference alleles, let's say, in that person's genome. And these bolded nucleotides here represent those. Then, to rule out the variant as one that isn't novel, or rule out the variant as novel, you compare those variants with those found in a panel of reference genomes, right? So in this example, you can see that this variant isn't likely a candidate for this person's idiopathic condition because that variant is seen in other people's genomes and those other people don't have that condition. So you're assuming that the variant is highly penetrant. However, there might be a variant that you don't see in other people's genomes, and you could infer that that is a logical candidate for the pathogenic or causative variant. And if it lands in a certain functional element in the genome, that you can probe with these computational tools and find out that it likely damages the functioning of that element, then you have more evidence in favor of that variant being the pathogenic or causative variant. So again, you start out with all the variants, eliminate those that are not novel, ask of the ones that are novel whether they are likely to damage a functional element, ask other sorts of questions like, does the gene that is harboring that variant participate in some pathway of relevance to the condition? So is the gene harboring the variant actually expressed in the gut? If it's not, then clearly it couldn't explain Nicholas Volker's condition, say. Ask if there might be compensatory mechanisms. And after kind of going in this reverse telescope fashion, you end up with a few candidates to the pathogenic variant, right? But there are some real questions here. What bioinformatics tools should be used to assess functionality, right? Does it make a difference, right? What reference populations, and this is where I want to go with the bulk of the upcoming slides, for determining the novelty. What do you use? What is that panel of patients? Does it matter who you use? Okay, shifting gears a bit, here's a depiction of a gene and the functional elements surrounding a gene. So I got this from book chapter. I helped it in Barnes a number of years ago. It turns out that with computational tools alone, you can more or less predict how nucleotide substitutions in these different genic elements might impact the functioning of that element. Now, these tools aren't perfect, but for example, you can make predictions about the likely functional effect of amino acid substitutions in a protein using conservation criteria, structural information, et cetera, et cetera. You can, using, again, conservation information and motif analyses and other tricks, make claims about whether a nucleotide substitution in a transcription factor binding site is likely to influence the strength of binding proteins to that site. So you can make these claims. So these are functional elements for which tools exist to predict the likely functional effect of nucleotide substitutions, right? And my group over the years has kind of contributed to this literature. We're not alone in this space. There are many outstanding groups working on computational tools, but we've published quite a bit in this area. Okay, shifting gears yet again. We worked with Complete Genomics, which is a whole genome sequencing provider up in Mountain View, California, to sequence the entire genomes of people from different global populations. So we could kind of survey how diverse the genomes were from individuals taken from different parts of the world. So this is not dissimilar from like the 1,000 Genomes Project and other stuff, only we cast a little bit of a wider net, and we wanted to sequence the genome deeply, not just rest content with this low coverage sequencing stuff. So we could really know just what variants populate individuals' genomes from these different communities. So what we did was work with, again, Complete Genomics to identify all the derived, that is, non-chimp alleles, and there's a reason why we did that, and I'll explain in a minute, in each one of these individuals' genomes. Then we subjected them to those computational tools that we had at our disposal and that we had developed to predict which one of these non-reference alleles that people possessed were likely to be functional and damage the functioning of a particular element. And then we could compare the total number and rates per genome of these functional variants across different functional categories. The number of likely transcription factor binding site disrupting variants in genomes from individuals of Africa versus Europe. The number of non-synonymous C-SNPs that likely damage the functioning of a protein between people from Asia versus those from Italy. We could address those sorts of questions. Okay? Then we could ultimately ask whether what we've learned from this the study of the diversity of the functional content of human genomes, whether that could impact the use of this kind of filtering strategy to identify pathogenic variants in the strategy that I described. Now, why did we look at non-chimp variants as opposed to non-reference genome variants? Well, some of you could probably guess why. The human genome reference is made up of DNA from people of European descent. So it's not a good comparator genome for people from the rest of the planet. Instead, you'd want a genome that could really be a comparator for everybody on the planet. And that would be an ancestral genome like the chimp genome. So we determined all the positions in each one of these individuals' genomes that deviated from the chimp genome, where there was both a derived allele, that is, an allele that was not present on the chimp genome, and one other allele possessed by one of the other people that we had, whose genomes we had studied, that was consistent with the ancestral allele. So the variants had to be segregating in the population. They couldn't be fixed for the derived allele, right? And they couldn't, of course, be fixed for the ancestral allele either. We were interested in the ones that were segregated. And then we labeled all those as likely functional and likely non-functional. Okay, and this was just recently published in Frontiers in Genetics. So here's what we learned about the diversity of these genomes. And some of this is consistent, again, with what you probably know about population genetics and ancient historical migratory patterns out of Africa hypothesis and whatnot. So I've kind of categorized these. These are the 4 African populations where we sequenced the genomes of individuals. These are 2 European populations, 2 Asian populations. These are kind of messed up a little bit. These are the Chinese and Japanese. And these are 2 admixed populations, an East Indian population and a Mexican population. The first thing you can see is this is just the total number of derived allele genotypes seen per genome. So in African genomes have roughly 6 million derived genotypes, alleles, loci that possess alleles, where one of those alleles differs from the chimp genome. But there are other people in the population that have the chimp genomes. Okay? There's substantially fewer in non-African populations. Right? And that makes sense because we know Africa is the oldest and most diverse population on the planet, so we would expect to see a greater diversity of genomic a greater diversity of the genome of people from those populations. Now, when we labeled those variants as likely damaging or not damaging or functional or non-functional, we then saw some patterns. So here is the number of likely damaging non-synonymous C-SNPs in these individual genomes. And by virtue of the fact that there's simply a greater number of derived genotypes in the African genomes, there's a greater number of likely damaging non-synonymous C-SNPs on the average African's genome, right? So about 2,800 or so in African populations and about 2,500 or so in non-African populations. Now, what's interesting, though, is when we looked at the number of homozygous derived genotypes, so not just the presence of a derived allele, present in a homozygous state, and we saw the opposite. In fact, despite the fact that there's a greater number of derived variants in the African genomes, in non-African genomes, there's a greater number of positions that are homozygous for the derived allele than in Africa. Right? Now, why is that the case? Well, it has to do with all sorts of effects of the bottleneck phenomenon and the fact that when people migrated out of Africa into the Middle East and then upwards into Europe, only a small fraction of the people present in Africa at the time actually made their way into the Middle East. Not everyone who was populating Africa at the time moved to the Middle East. And then a subset of the people that were populating the Middle East ultimately made their way up into Europe. So we wouldn't expect to see a couple of things. As great a diversity in the European and Middle Eastern populations than we would see in Africa. And 2, because of the smaller founder or effective population size in the Arab and European early communities, there's a greater chance for people to mate that are related to each other at some level. It doesn't have to be siblings. It could be third or fourth cousins. But that probability would be greater in populations where there's a smaller founder size, right? So think of it. You just have a finite number of mates to choose from. You're likely to choose a mate that you're more related to in a smaller population than if you have a big monster population. So that would lead to a greater frequency of homozygous genotypes, right, that carry these derived alleles. Now, as a result of there being a greater frequency of derived homozygous genotypes in the non-African populations, there's actually a greater frequency of functional derived homozygous genotypes in the non-African populations. Now, this has actually been documented to some degree in the literature. Carlos Bustamante and colleagues published a paper kind of arguing that this was the case using some empirical data in just a few populations and looking at a subset of all the genes that populate the human genome, and basically showed something very similar. But they didn't use whole genome sequencing data. And they didn't use an array of computational tools to consider functional elements of all types that populate the genome. They only looked at coding variants. Okay, so this is kind of an eye chart, but I think I can walk you through it. So this now focuses on the variants that were unique to each one of the populations we studied. And here we lumped the populations into kind of continental categories. So we had African, European, and Asian populations. So we lumped the people together into these categories. And these are the rates per genome of variants that are only found in those continental populations and not the other ones. So these are population-specific derived alleles. So in the African populations, you can see that there's actually fewer population-specific variants, right, than in the non-African populations per genome. Okay? So, if you just focus on this, you see that these rates are uniformly larger in the non-African populations. So this means that if there is a variant that you only see, and these are all functional variants, by the way, I should have emphasized that. That means that if you find a variant that is only present in European individuals, right, it's more likely to be functional than in the African populations. But that's confined to alleles that are unique to those populations. Okay? And these are highly statistically significant. These are the p-values comparing Africans and Europeans and Africans and Asians. You can see it's highly significant, these rates per genome. This had never really been shown before. Now, what does that have to do with that filtering strategy? Pretty obvious. I think if you just follow some of the logic I've been trying to impart to you guys. If you have a patient who has an idiopathic condition, and you want to rule out variants as novel in that person's genome, you have to use a reference set of genomes that is matched for the ancestry of your patient. Because we know that the functional content of the genomes varies widely from population to population. And some alleles that are unique to populations, right, that are functional, also differ widely between populations. So in other words, if you had a European patient and you wanted to follow this filtering strategy, but your reference population was made up of African individuals' genomes, then you would be misled to a much higher degree as to what the likely pathogenic variant is than if you used an appropriately ancestry-matched population like Europeans. Okay? So population genetics and variation in the frequency of functional variants on a global scale has very important and practical implications in the clinical use of genome sequencing. And this is just another example where this is kind of the false lead rate, the number of times a researcher might be misled with respect to coming up with a likely pathogenic variant if they used an inappropriately matched reference panel to make claims about what is novel in the patient with the idiopathic condition's genome. Okay? This is another little eye chart, but this basically says the same thing. So these ratios here are the number of false leads per the number of variants that you might consider as candidates for your idiopathic disease condition. Where if you had a patient that was either European, African American, Mexican, right, and then compared that individual's genome to reference panels of different sorts, say the 1,000 genomes data, or this panel of complete genomes that we have with complete genomics. Here's the number of times, again, that you would screw up. So if you used an appropriate reference panel, say European reference panel for a European individual, you'd have about 194 potential false leads, where if you used an African reference panel, you'd have about 680. And that's using 1,000 genomes data. And you can see that kind of similar patterns of hold with different data sets. Now also keep in mind that the number of genomes in these reference panels is highly different. So only 13 to 20 or so with the complete genomics panel, because that's all we had at our disposal. Whereas, they've sequenced upwards of 500 individuals in each one of these populations, 1,000 genomes. So the first point, which I had emphasized before, is genetic background of the patient in the reference panel make a huge difference with how often you might be misled as to what is a causative variant for an idiopathic condition. Also, the sequencing platform makes a difference. I didn't go through this in great detail, but it's just present in the slide. So whether you use the complete genomic sequencing technology or the Illumina sequencing technology, as was used by the 1,000 genomes, it makes a difference. That is, these numbers are actually different. So clearly, there are differences in the reliability of variant calls using one sequencing technology or another, or a different pipeline for calling variants. So you have to be careful with that. The other thing, and again, I didn't go into this in great detail, the functional analysis or computational tools that you use to label the variants as likely functional makes a difference. So there's two tools out there to predict the likely functional consequences of amino acid substitutions, non-synonymous C-SNPs. There's a program called Polyphen, and there's a program called SIFT. They use slightly different strategies. So you can see that the rates, the false leads, are actually different depending on how you label the variants as functional or not. So that's another take-home message. But the real one had to do with the diversity. The human population is made up of a very diverse set of humans, at least at the genetic level. And we see substantial differences in the rates of functional variants in people that populate different parts of the planet. Okay, so now shifting gears, going to the imaging stuff. As you probably know, there have been reports purporting to have identified variants that influence kind of brain size. So this one from Bruce Lahn's group generated a reasonable amount of controversy, 7 to 8 years ago, where they had published a report saying that variations in ASPM actually influence brain size. And the frequency of the variant that influenced brain size varied dramatically from population to population. So in their report, they had examples of populations with a high frequency of this variant that tended or was associated with larger brains. And then they found populations that had a low frequency of this allele. Now, what they didn't do is go out there and say, well, this explains why some populations might be smarter or not as smart as others. They didn't go do that, but other people interpreted their data as this, and it generated a lot of controversy. But I think no one in this room would deny that there aren't genetic influences on brain morphology. It's just how you interpret those or use that information for whatever purpose, which is controversial. But at any rate, they had purported to identify a variant, segregating variant in the human population that was associated with brain size. Now, we got interested in this because there are certain diseases where brain size is thought to influence or be part of the pathology, and autism is one. So for many years, I was associated with an Autism Center of Excellence in San Diego, led by Eric Corshane and colleagues, no longer a part of that. The grant was not renewed. But his belief was that one of the hallmark features of autism was an early brain overgrowth, that there were children that during certain developmental period had a larger increase in kind of brain size. It then slowed down after this period, but he thought this was a hallmark pathology of autism. So then the question is, well, were there any naturally occurring variants that might have influenced that early brain overgrowth and were associated with brain size? And I won't go through the facts about autism, but you know it's a devastating disease. Its epidemiology is becoming a lot more clear. Now, we studied a particular gene, MECP2, because MECP2 had been studied by other labs and was known to be an incredibly potent transcription factor on the X chromosome. And in fact, in this little figure taken from this paper, These color codings here just represent the number of genes that were up or down regulated when MECP2 was tweaked in certain ways. So if you inhibited the expression of MECP2, all these other genes, expression levels were changed as a result. And if you increased the expression of MECP2, then a whole bunch of genes had their expression change as a result. So one tweak in a gene could upset the functioning of an entire network of genes. And so you could imagine how if there were inherited variations in MECP2, because it's a potent transcription factor, it could influence the functioning of many genes that might impact brain size. So we thought this is a good candidate gene. So here's just an example of a paper that we published with Eric and his group, looking at this overgrowth phenomena using imaging techniques. So this is all published, and some of you may know. This is not, accepted by the community. There's a lot of holes in the argument, but the data are what they are. So you can see that we had these MRI scans done on autistic and non-autistic children at very early ages, and we had longitudinal data, so we could see that, on average, there was an increase in brain size and growth in the autistic versus the non-autistic kids. Okay. So then what we wanted to do is say, okay, we want to study autism not only with respect to autism, which is our main target, but just consider naturally occurring variations in that gene and their association with brain size generally. So forget about one condition, just ask if variations influence brain size in the general population. So we worked with Anders Dale and Ole Andreasen from Oslo. We had an initial data set, had about 300 subjects with both genotype data and structural MRI that Ole had collected as part of his top study. Some of these people had pathologies, roughly 50-50 males and females. We looked at this MECP2 gene. This paper was published 3 years ago or so. We also had replication studies that we pursued with the ADNI data that I know you're all familiar with. So here's just a table describing what we found, and this is right from the paper. These are different SNPs in the MECP2 gene, and here are p-values with their association strengths for different parameters derived from the imaging. So intracranial volume, brain volume, cortical area, cortical thickness. Right? Here is our follow-up study and their p-values in the ADNI. Now we didn't have the same number of SNPs because the chips that were used on these populations were different, and imputation techniques as little as 4 years ago aren't what they are today, so we didn't pursue those. So you can see that in the initial study, the top study, we saw some pretty strong associations, even after correcting for multiple comparisons, subsets of which looked like they replicated in this independent data set. And Anders has kind of developed these strategies for depicting, as of other groups, the strengths of associations globally on brain structures. So the more red the region, the stronger the association between the MECP2 variants in that brain region, and the more gray, the weaker the association. Okay. Yeah.
Quick question: are those variants functional or just around, are those coding, non-coding, or?
They're not all coding by any stretch, and of course you can go through and see, but no, some of those associations with variants whose function is entirely unknown, and we've subjected them to our computational tools. Many were predicted to be neutral. So why they're associated, we don't know. It could be that there is some unknown enhancer or silencer in the region that's being tweaked. We don't know. But no, that's a good question. So at any rate, we got on this kick that, well, maybe we could find other variants that influenced kind of natural variation in brain size by melding genetics with at least MRI technologies. So we went back and looked at genes like ASPM that Bruce London saw in these same populations and in the ADNI populations. And this was another paper that was published, just looking at genes known to influence microcephaly and other phenotypes, and looking at whether or not variations in those genes influenced general brain size variation. And found some evidence that they do. Subsets of these SNPs and the genes that they sat in were replicated. And again, we can come up with these nice looking diagrams of what brain regions appear to be influenced most by these SNPs. So that suggests that there may be naturally occurring genetic variations out there that influence brain size, right? And that may be of relevance to disease. Just to let you guys know, we did sequence a bunch of people with autism and MECP2 and found all sorts of deletions and stuff, but we didn't have a big enough sample size to sort of get p-values low enough. So that study is still ongoing. But what really motivated MECP2 was the fact that variants in MECP2 had been associated with Rett syndrome, all the other information I had said. And just as a byproduct, we thought, well, we'll just look for variation in MECP2 and its association with general brain morphological variation. So because I'm interested in population genetics and wanted to use that initial study on idiopathic disease sequencing to kind of expose you to my interest in population genetics, we've got an interest in looking at how frequent variations are, much like Bruce Lawn, on a global scale that influence brain morphology and what they might mean in the grand scheme of things. Okay? So we published this paper a year or so ago where we wanted to look at the relationship between genetic background, ancestry of individuals, and brain size, parameters derived from MRI. So our hypothesis was that brain size, as with skull size, shows variation consistent with historical migratory patterns in at least Europe. Okay, and we'll show you data later where we looked at this at a global scale. Now, it's well known that craniometric features vary widely on a global scale, right? It's almost undeniable. The data are overwhelming. People's skulls are bigger, smaller in different populations. So then the question is, why wouldn't what's inside the skull also vary, right? So since we had MRI and technologies and whatnot, we could look at this. So we thought this would be interesting and kind of fun to explore. So we had two data sets for this paper. We had skulls obtained from individuals living in different European communities that were available from public domain data sets from museums and whatnot. So we knew where those people were sampled from, and we could get these measures on the skulls that are in the public domain. We also had brain images from individuals whose ancestry could be determined using genetic methods. So it's all possible with enough genetic markers to kind of label someone as likely from Northern Europe, or likely from Southern Europe, or likely from Belgium, or likely from France, all these sorts of things. I'll show you how that was done in a minute. So our basic analysis strategy was to correlate skull and brain size with these major sources of population and individual genetic differentiation that might reflect migratory patterns. I'll describe how we did that in a minute. So, as I talked about in the context of the idiopathic disease sequencing study, migratory patterns are known to have come about in certain ways. In Europe, there is a migration from the southeast to northwest, a historical migratory pattern. Okay? And we know that there is substantial morphological variation, including cranial variation, among people that populate Europe, with the Norwegians having kind of the tallest, being the tallest people, having the biggest brains, and people from Spain and other communities having, on average, smaller frames and smaller brains. And there's tremendous kind of craniofacial variation and differentiation on a global scale. So the data set that we got looking at the skull or cranial variation had been studied by different groups. Robert Sokol and others had published papers where these little dots represent where they had skulls from, and they showed that the closer the two skulls were, that is, the closer two people lived whose skulls were used in this thing, the more similar their craniometric features were, suggesting that there was a pattern. Somehow people living closer together or in the same community had similar craniometric features. Okay? We reanalyzed the data. We got a much larger data set. So here's where the skulls were derived from that we had. So we had about 1,200 male skulls from 26 different populations. About 37 different kind of cranial and facial length measurements were made. And about 85% had complete data. And here's what we found. We also found a trend for people whose brains whose ancestries were very similar. They lived in the same communities, had similar craniometric measures. So this is the correlation between the geographic distance between the two skulls in terms of where they were taken from and their similarity. Now, many years ago, we published a paper describing a technique that could be used to relate kind of similarity in certain features to genetic background, called the generalized analysis of molecular variants, because there was a tool that was proposed in the literature before by Laurent Escoffier and colleagues called the analysis of molecular variants. We just said we've come up with a technique that kind of generalizes that. And we actually went back and looked at some historical skull data with some actual SNP data from people in those same communities, and basically found that the more similar the genetic background of two individuals, the more similar their cranial features. And here are little trees showing that. If you just clustered the people into groups by either their craniometric similarity or their genetic similarity, you saw a strong correlation between the two. So what we were able to do is use a strategy like principal components and multidimensional scaling to pick out the sources of variation that contributed most to the craniometric measures that we had. And we found that one major source of variation was consistent with this ancient or historical migratory pattern from the southeast to the northwest. So you can explain a lot of the brain variation on the basis of some historical migratory pattern. So we had for each brain, or each skull, where they sat on a line that connected the southeast to the northwest, and then asked if that was correlated with the craniometric features. And in fact, for many of these parameters, it was. Suggesting that you could explain some of the craniometric variation on the basis of this historical migratory movement from the southeast to the northwest. And it might have to do with genetic drift, with environmental exposures of people in the northeast to the southwest, et cetera, et cetera. Now, we had this other data set where we had the imaging data, the brain imaging data. Right? So there we could explain skull differences on the basis of this ancient historical migratory pattern. Now, with this other data set, we're going to say, well, is the brain variation, does it follow a similar sort of pattern? We got people from the TOPS and ADNI study, and then using the genetic markers that they sat on, lumped them into positions along this migratory pattern from the southeast to the northwest. And we could do that using techniques that are probably known to many people in the room, principal components and other sorts of things. And Carlos Bustamante and all had a paper describing how in Europe, with enough markers, you could separate people into different communities. So we applied that same strategy to everyone in our study who had the imaging data. These are kind of the tools that we used, and we also had to control for things like age and sex, height and weight, importantly, as will become clear in a minute for the people, and then look to see what the strength of that correlation was. Here are some results. This is the intracranial volume, this is cortical area, and so you can see that there is a correlation of sorts between where a person was from, their genetic ancestry, along this northwest to southeast or southeast to northwest axis, correlated with intracranial volume and cortical area. Now, we didn't have so many people in the middle. There was a lot of people on the ends. It's true. And so some people said, well, if we just confined attention to those people that were, lying across the middle regions and not just at the two ends, what happened? And the correlation was still there, it just wasn't as strong. Okay. So this wasn't due to some big environmental difference between people living in the British Isles and people living in, communities near the Middle East. And we could show this for any number of parameters we could derive from the MRI data. Okay? And then we could rank the strength of the correlation between the different phenotypes and where an individual sat on this ancient historical migratory climb. When we looked at the brain regions that looked like they were most affected by this ancient migratory pattern, interesting things emerged. It wasn't just brain size overall. It was particular morphological features of the brain, suggesting that the shift in the brain wasn't global. It was different parts of the brain. Okay? That was the most interesting. So to kind of conclude here, for the skulls, we saw that a northwest to southeast or southeast to northwest axis of cranial variation aligns with the primary axis of genetic variation. Cranial length and breadth increase, and height actually decreases by about 5% along this northwest to southeast axis. For the brains, this was just one parameter, intracranial volume and cortical area increase, about 5% along this northwest to southeast axis, and that's independent of height and BMI. So there was some pull going on, whether it was drift, genetic drift, or something else, selection, who knows. And there was a definite posterior anterior shift in cortical areas along this northeast or southeast to northwest axis. And so this ultimately suggested that it's more subtle than just having bigger brains equaling bigger skulls and vice versa. Okay, so I'll shift a little bit and I guess I'll whiz through this. There's a lot of interest now in deciding whether a trait can be attributed to the combined effects of many variations in the genome, or just a couple of variations that have a more pronounced effect. Most traits have some polygenic basis to them. The question is whether or not there are genes with larger effects that work over and above them, or whether it's all due to polygenes. There have been many papers published in the literature, including a lot recently by Peter Vischer and colleagues, showing that you could actually estimate the degree to which a trait is influenced by polygenic factors versus a couple of SNPs or whatever with big effects using GWAS data. So this is not just estimating heritability for twins, it's actually using genotype data. Okay? So essentially you take a phenotype and you regress it on the phenotype and the genotypic similarity of the people in your study. And if there's a correlation, then you can attribute that to the combined effect of many variations, because your measure of genetic similarity is across all variations of the genome, not just one or two. That Gamova paper that I talked about before kind of did the opposite. Here you regress kind of genotypic similarity on some factor, which could be brain size, it could be whatever, and it's been shown in the literature that these two strategies in certain situations are more or less equivalent, algebraic. Won't go into that. So our group has been interested, as I had mentioned, in using imaging technologies to sort of get at potential intermediate phenotypes for disease. And so we've published a few papers over the years. So we're not just interested in population genetics, but really understanding how genetic variation influences brain morphology in a way that might shed light on certain disease states. So the PING study is a large study, pediatric imaging, neurocognition, and genetics study. It's focused on comprehensive neuroimaging, cognitive measures, and GWAS data in a very large pediatric cohort that is sampled from all populations, all global populations. So it's large, about 1,200 participants with very diverse ancestries. This is kind of a depiction of how diverse those ancestries are and the degree of admixture exhibited by the people that are participating in the PING study. So red is European, so we have many people that are almost purely of European descent. Many individuals that are admixed, so maybe African Americans, where they have some degree of European ancestry, but the bulk of their ancestry is of African origin. We have Hispanics, we have people from Oceania, other populations. So we have a tremendous diversity among the people in the PING study. So what we did was just correlate the degree of admixture exhibited by these people with the brain phenotypes, right, that we could get out of the MRI. So if this is a plot of the degree of European ancestry exhibited by individuals in the PING study, correlated with total cortical area, you can see that there is a positive correlation, and it's quite strong. Now, we did have many individuals that were either purely European or non-European 100%. Right? But we could also turn this around. Instead of asking, well, what is the correlation between the degree of European ancestry and total cortical area, we could ask, well, what is the correlation in the degree of African ancestry and total cortical area? And you can see we got some highly significant correlations. when we got rid of the people at the two ends to kind of argue against this correlation as being a function of some environmental exposure of Africans versus non-Africans, right, that is driving this correlation, we still see the correlations there. And this is just summary tables of different parameters, what the ultimate measure of admixture we're looking at is, whether it's degree of European admixture, African admixture, et cetera, et cetera. But we see correlations, and this paper is being written up by a postdoc in my group. So these correlations are likely due to some kind of polygenic effect. And why do I say that? Well, let's say there's only one or two genes that are actually influencing brain size, right? Now, you wouldn't expect to see the degree of admixture shown by people correlate with brain size in quite the way that we do. Instead, it looks like there are many, many different genes that are contributing to brain size. It's a polygenic effect. So the degree to which you have polygenes from one population or another is going to impact your overall brain size. Okay? So this might explain other things about morphological differences and brain morphological differences between people on a global scale. Okay? Actually, if we look at kind of growth trajectories, which is really what the pain is all about, and you lump people into different categories, you can see that the trajectories are, in fact, different. So there's some different developmental pattern that is probably influenced by the combined effect of many genes in these populations. Now, what does this have to do with kind of using MRI as diagnostic tool? Well, let's say you have a person who comes in, and maybe you want to see if you can't determine if they're having an early brain overgrowth that's consistent with autism. So you're going to subject them to an imaging protocol to see if their brain is bigger than it should be. And the question is, well, what are you going to base the should be on? Is it bigger than what it should be? Well, if you have an inappropriate reference population, you're screwed, right? So if you have an Asian individual that you're trying to evaluate for how big that person's brain is, and your reference population is Europeans, and we know there's this tremendous global variation, then all bets are off into what quantile you're going to lump them into that might be indicative of small brain versus large brain. So that much is pretty obvious. So here's the real question. What do you do with the admixed people? So they don't necessarily fall into any category. You're going to have to understand the degree to which admixture actually influences brain variation before you can come up with the appropriate norms, right? You can try to regress everything out, but because the trajectories are actually different, how do you control for that? So this presents a real problem. And this is not unique to use of brain imaging. You can imagine blood pressure variation, right, across a global scale, where you have someone in Africa, and maybe Africans just by and large have more elevated blood pressures, or Norwegians have more elevated blood pressures because by and large they're a larger people, right? Then you need to develop the norms for blood pressure in those different populations. And that much, I think, is pretty much obvious, and it's probably attributable to the combined effects of many genes and polygenic effect. But then the real question is, well, how do you evaluate an admixed individual? Do you have to be appropriate ancestral reference populations, and do you use a linear combination of them to come up for, what do you do? So, okay, last couple of things I'm going to whiz through here are some other studies where we tried to combine genetics with imaging data to address questions that we thought were interesting. So here's a paper that was published a little while ago where we looked at the association of genetic variations with the scaling of the visual cortical surface in humans. So our interest was not in whether a variation influenced a particular brain-derived parameter. It was actually whether a SNP influenced the relationship of two imaging-derived parameters. So was there an interaction between, say, a SNP and one imaging measure as they influenced another imaging measure? So here we wanted to assess the impact of genetic variations on the relationship between the occipital cortical area to total cortical area. So we had occipital cortical area written as a function of a SNP, total cortical area, and then a total cortical area a SNP interaction. And this is what we wanted to see, whether there was a significant result with any one of these SNPs. We used the top study, and it was GWAS. Here were the results. It was a QQ plot suggesting that we had some evidence to believe that a SNP had a low enough p-value to convince us we might have a real association. Here is the area of the genome. It was right in this gene, GPC PD1. where there was a variant associated. Here's kind of the Manhattan plot. So it sits right here, so it looked fairly significant. Here's sort of a table. Oops, here's a table describing the results, the association in different studies of the most strongly associated SNP with different phenotypes. Here's just a plot suggesting that this really was an interaction. So if you plotted the occipital cortical area as a function of the total cortical area, where you lump people into the SNP categories, you see that the relationships are different. So this is what was actually significant, the p-value of 10 to the minus 9, suggesting that this SNP influenced the way in which occipital cortical area was influenced by total cortical area and vice versa. Okay, then the last thing I'll tell you about, we also studied in this kind of interaction context the influence of SNPs on imaging-derived parameters in schizophrenia. So our hypothesis was that cortical thinning, which is often observed in people with schizophrenia, is exacerbated by SNPs among schizophrenics, or the effects of SNPs on cortical thinning is exacerbated by the schizophrenic environment. We didn't know which one, but the idea was, again, to search through the GWAS data for certain interactions. So we said schizophrenia could be a function of cortical thinning, some SNP, and then an interaction. So there might be a SNP that exacerbates that cortical thinning and induces schizophrenia. So we were looking for interactions like this. Again, we had a big data set and did this. Again, had p-values that looked significant. These kind of landed, the most strongly associated SNPs landed in a region of the genome harboring Prader-Willi and Angelman syndrome genes, which kind of made sense from functional standpoints. So I'm not going to be able to go into great detail with that. We had 94 schizophrenics, people with different conditions, bipolar disorder. We had control individuals, and you can see that the SNP was most strongly associated with the schizophrenic, suggesting that this interaction was actually reflected in the data. So you can see no difference here, it's only in the schizophrenic. Okay. And then we could again plot what regions of the brain looked like they were most strongly affected, and we actually showed that the SNP also influenced a few other kind of neuropsychological phenotypes that we had. So in conclusion, whole genome sequencing is here, as are other genome-wide assays. Interpreting genomic variation is the impediment to genomic analysis, not generating the data. I think that's safe to say. Human diversity needs to be considered in evaluating the uniqueness of genomic elements for all the reasons I described in that study of the identification of pathogenic variants. Human brain morphology varies widely across global human populations. I think this is undeniable to everyone in the room. The question is, what is driving that? Is it genes? Is it environment? Is it a combination? Brain morphology appears to have a polygenic basis revealed through admixture studies. So those correlations between degree of admixture and brain morphology, I would argue, suggest that there's a polygenic basis to brain morphology, though someone may have anticipated that, just knowing a little bit about all the different neural circuits and whatnot that genes have a role in mediating. Genetic variants may impact brain morphology in ways that ultimately contribute to disease, like the schizophrenia study. So I'll just thank my colleagues here and students and then the grants that support them. So thanks, I know I went a little over.
I'm going to take some questions.
So this is a general question. So apart from brain disorders, so can you say that there is a consensus between skull and brain size and cognitive function? Let's say IQ or some other global measure of cognitive
function. So cognitive function, brain size. So the Ping data would allow one to address those questions. I can tell you that the Ping collective has been sort of reluctant to make these sorts of claims for all the politically charged reasons that you could imagine, right? I have been kind of a defender of this idea that it makes perfectly good sense to me that genetic variations influence brain morphology, because everybody knows that genetic variants influence just about every other morphological parameter that we can measure. Skull size, arm length, you name it. So why not the brain size? And to me, it's not as taboo as people think. But when we start linking neurocognitive abilities, IQ, other things, then we run into a little bit of trouble. But I'll even go out on a limb and say, I think it's undeniable that genetic variations influence IQ. if you have multiple copies of chromosome 21, it's been shown that your IQ is on average much lower. You have Down syndrome, these things are obvious facts. The degree to which polygenic background influences IQ and neurocognitive abilities and whatnot is an open question. And we'll probably learn a lot about that. Now, whether that should be seen as taboo or not, I don't know. I mean, I have my own opinions on it, but we're going at this kind of carefully.
This is going to betray my lack of knowledge here, but variation in brain size could in principle be due to variation in number of neurons or something about their size or packing arrangement. My question is, does the answer to that question vary in different populations?
Well, that'd be a great question. So, if we actually had measured this, Eric Courchain and others have phenotypes that they're looking at with respect to autism, which is a number of neurons, number of, and making the argument that these might be hallmark pathology diseases. So if they could measure them in disease states, you could certainly measure them in normal, quote-unquote, populations and then see if genetic variations influence those, and not just kind of these gross phenotypic measures that we get from MRI. So that's a logical thing to address. We just don't quite have the data to look at that, but I think it's a great way to pursue these kind of future studies.
So you mentioned that brain morphology is changed on partially controlled by the genetic features. And what's the effect size compared to usually we do patient versus controls and looking at the morphology differences? Right? What's the comparison, the effect size?
Well, it's not huge. I mean, when you looked at those regression lines, it wasn't like this. So, the percentage of variation in these brain morphology phenotypes that we look at is actually quite small. So the R-squared's on the order of like 2 to 7%. So that means clearly there are other factors that influence brain morphology, and those could be environment, diet, you name it. We just don't have the detailed information to say precisely what the contribution of each one of these factors is, but we're getting there, I think is the point. But my point is that genetics is a role that's undeniable. Doesn't mean it's the major player, but you can't deny it.
If the case where the effect size is quite small compared to patient controls, then when we do a typical patient control comparison, maybe results is not that critical whether we consider genetic or not.
Absolutely. So if in fact those effect sizes are not so pronounced between, say, Asians and non-Asians, maybe this concern about having an appropriate reference panel for Asian brains is not that big of a deal. I fully agree. It might be the case. I think we just have to do the right studies and see if it does make a difference. Like, how many people would we falsely label as likely autistic on the basis of brain size? It might be very small, 2 to 5%, and maybe that's okay from an epidemiological or public health standpoint. I don't know, but until we put it to the test, we won't know.
Thank you.